My name is Megan Schroeder, and I am the Assistant Section Head for the Molecular Diagnostics Section at the Texas A&M Veterinary Medical Diagnostic Laboratory. My presentation today is on Trichomonas fetus testing at the Veterinary Diagnostic Laboratory, with a focus on PCR testing and how it led to the development of improved molecular diagnostic testing reagents for PCR detection of T. fetus. Trichomonas fetus is a sexually transmitted disease of cattle caused by the flagellated protozoan parasite Trichomonas fetus, or more commonly known as T. fetus or trick. T. fetus infections in cattle can have a significant economic impact for farmers and ranchers due to several factors such as decreased calving rates, extended calving seasons, culling and replacement of infected cattle, and maintenance of non-productive cattle, which can include costs associated with feeding as well as diagnostic testing. There are several clinical signs associated with trick infections, including abortions, infertility, and various reproductive issues. Most clinical symptoms generally clear within several weeks or months of reproductive rest. However, permanent infertility is possible. Bulls are considered asymptomatic carriers of infection and are thus the focus of regulatory and diagnostic testing. Diagnosis of trick involves collection of a preputial smegma sample that is then submitted to the diagnostic laboratory for testing by either culture and microscopic examination or real-time PCR. The gold standard for trick diagnostics is culture followed by microscopic examination. Preputial smegma is placed in a pouch containing culture media that promotes growth of the T. fetus organism. The pouch is incubated at 37 degrees Celsius for up to 7 days and observed daily for the presence of motile trick organisms. While this method is a low-cost, quick and easy tool, it does present some challenges in terms of sensitivity and specificity, particularly in its ability to distinguish T. fetus from other closely related species. With improvements in science and technology that led to the development of PCR, and specifically real-time PCR, diagnosticians now have a diagnostic tool available for testing that provides both a sensitive and specific method for detection of T. fetus. While samples are still submitted in culture media pouches, incubation times are shortened and samples can be tested with results reported in less than 3 days. In addition, validation studies have demonstrated that samples can be pooled up to 5 total samples for PCR testing, which in turn provides a cost savings for clients. Many states throughout the U.S. have trick regulatory programs that require testing of bulls prior to movement or sale. While trick testing volume varies at different veterinary diagnostic laboratories throughout the country, depending on lab location, at TVMDL, we test upwards of 40,000 trick samples annually. While we receive a decent number of test requests for trick culture, the bulk of the testing requests are for PCR. Rules and timeframes for sample submission vary by state, but per our Texas Animal Health Regulatory Commission, bull preputial smegma should be submitted to the diagnostic laboratory within 96 hours of collection. Submitted samples are required to be incubated at 37 degrees Celsius for 48 hours, whether at the veterinary clinic or the diagnostic lab prior to PCR testing. With the volume of trick samples we receive annually came several challenges with cultured samples for PCR testing. These challenges included variations in sample temperature and time in transit before arriving at the laboratory, the burden associated with incubating the cultured samples, both for the veterinarian collecting the sample as well as the laboratory, the potential for bacterial overgrowth in the cultured samples, which can inhibit detection of trick, costs associated with purchasing and storing the culture media for long-term use before expiration, and lastly, the challenges experienced with processing these samples at the laboratory. These challenges can be both time and labor intensive for sample processing, thus adding to the turnaround time before results are available to the client or veterinarian or submitter. As a result of these challenges, our molecular team began working several years ago on a solution to simplify the testing workflow. Our improved workflow utilizes direct smegma from bovine samples without the need for sample culturing. In terms of sample processing, it provides several benefits to both veterinarians in the field collecting samples, as well as technicians processing these samples in the laboratory. In regards to sample collection in the field, bull smegma is collected into a tube containing approximately 2 mL of phosphate buffered saline. The sample is kept cold and shipped to the laboratory, preferably on ice packs. Since culture media is not used during the collection process, there is no need for sample incubation, either prior to shipment by the veterinarian or submitter, or at the laboratory. Once the sample is received at the laboratory, as mentioned, there is no need for the use of culture media, thus no need for sample incubation. The lack of culture media combined with the lack of sample incubation not only results in less burden on laboratory technicians for sample processing, but also leads to a quicker turnaround time from sample receipt to results reported. We frequently receive questions in regards to sample collection for submission to the laboratory, so we have provided suggested sample collection guidelines here, which can also be found on the Texas Animal Health Commission website. In brief, we recommend smegma samples be placed in 4 ml tubes containing approximately 2 mL of phosphate buffered saline, or PBS. In order to ensure that all smegma is deposited in the tube, the collection pipette should be rinsed several times by aspiration of the PBS in and out of the collection pipette. Once the smegma sample is placed in the tube, it is best to keep the sample cool, preferably by storing with ice packs, for shipment or transport to the laboratory. And of course, for identification purposes, we recommend the tube be labeled with the bull ID and collection date. Once samples are received and accessioned at the laboratory, they are then ready to be processed for testing. The next few slides here will describe the sample processing workflow once the sample or samples are received in the laboratory. Processing of the samples occurs in a two-step workflow. The first step of the process is the extraction of nucleic acid, which is shown here. The direct smegma sample is used for nucleic acid extraction with a commercially available kit. All sample extractions include the use of an exogenous internal control, which is added during the extraction process. The use of this internal control allows us to monitor the extraction and PCR processes, as well as monitor for sample inhibition. Following nucleic acid extraction, samples are tested by real-time PCR for the presence of T. fetus nucleic acid. Since culturing of samples and culture media prior to extraction is not required with the updated extraction workflow, we developed a PCR with improved T. fetus detection sensitivity. This improvement in the real-time PCR specifically targets the detection of T. fetus RNA as well as DNA. The PCR master mix contains a reverse transcriptase enzyme that amplifies RNA in addition to DNA, thus improving the sensitivity of detection. As with cultured PCR samples, any sample with the CT value less than 35 is considered positive. One of the benefits of PCR testing is the ability to specifically target the pathogen of interest, in this case, T. fetus. PCR oligo sequences, or primers and probe, are specific to the 5.8S rRNA gene and are not found in any other closely related organisms, including T. vaginalis, Tetratrichomonas gallinarum, Pentatrichomonas hominis, or Simplicimonus. And the validation data describing these improvements was published in JVDI several years ago, and the method patented, with the references listed here at the bottom of the slide. With TRIC being a regulatory program, TRIC testing has provided some testing challenges for veterinary diagnostic laboratories. Each laboratory has variable timeframes as to when samples should be submitted to the laboratory following collection, as well as different extraction methods and different PCR testing protocols. There have been many discussions over the years in various venues, including TRIC working groups, as to how laboratories can potentially standardize testing. While laboratories have yet to agree on a diagnostic protocol, we wanted to see if there might be a way in which the improved workflow, specifically the PCR reagents, could be provided or shared with other laboratories in order to help relieve some of the burden associated with trick testing, as well as potentially move closer to standardization of testing. Following the publication of the improved workflow, we had several discussions with Thermo Fisher about how we might be able to provide PCR reagents that would offer a standardized solution to diagnostic laboratories for the improved trick T-fetus testing procedure. In collaboration with several Thermo Fisher scientists, we further enhanced T-fetus PCR detection in direct smegma samples by updating and validating a newer PCR master mix chemistry. This master mix contains the reverse transcriptase enzyme in the master mix buffer, along with the previously published T-fetus primers and probe design. And this improved chemistry was then utilized for the validation of pooled direct smegma sample testing. The first step of this process was the verification and validation of the improved master mix, which is the TaqMan Fast virus one-step master mix. The graph here to the right shows the result of that experiment. CT values are labeled along the y-axis, sample IDs are along the x-axis. Approximately 20 samples were tested in triplicate with both the previously published reagents and protocol, as well as the newer master mix chemistry. As shown, the newer master mix chemistry provides slightly lower CT values and thus better detection sensitivity than the previously published reagents. The second step of this process was the validation of pooled smegma samples. Currently at TVMDL, greater than 40% of the trick culture samples that are submitted for PCR testing request sample pooling. Pooling of samples provides clients with a cost-effective tool for screening herds at low risk of trick infection. Therefore, we wanted to validate pooling of direct smegma samples for PCR testing. For the validation of direct smegma pooled testing, we utilized 100 trick-positive smegma samples and 100 negative smegma samples, as determined by our routine testing and diagnostic submissions. Samples were first tested individually to verify CT values, and then each sample was used to create a pool. For trick-positive pools, 1 trick-positive sample was combined with 4 trick-negative samples, and for trick-negative pools, 5 negative samples were combined. Each pooled sample was extracted and tested with the newer PCR chemistry, and the CT results compared to those of the individual samples. The results of the pooling validation are displayed in the graph shown here. Again, CT values are along the y-axis, and sample IDs along the x-axis. Blue dots represent the CT values for the samples tested as individuals, and the red squares represent the CT values for the samples when tested in a pool. As expected, the pooled sample CT values are approximately 2.5 CTs higher than the individual sample CT values, and still detectable by PCR. In order to adjust for this change in detection and to identify individual positive samples in a pooled sample, we determined an increased CT value of 38 as a cutoff for detection would be appropriate. In other words, any pooled samples with CT values less than 38 would be considered positive pools and subsequently be tested as individuals, and any pooled samples with CT values greater than 38 would be considered negative. These parameters are similar to the analysis parameters used for PCR testing of pooled culture samples. In comparison to PCR testing of cultured samples, detection of individual direct smegma samples has the same parameters as detection of the cultured samples. When tested individually, the CT cutoff for positive samples is 35. Any sample with a CT value less than 35 is considered positive, similar to cultured samples. When tested in a pool, direct smegma samples can be pooled up to 5 samples in a pool, with a pooled CT cutoff of 38, which are the same parameters as for cultured samples. Any pooled sample with a CT value less than 38 would be considered positive, and just as with cultured samples, would require testing of each of the individual samples within the pool in order to determine which sample or samples are positive. As mentioned earlier, and as with all cultured samples, all smegma samples tested either individually or in a pool contain an internal control in order to monitor for sample inhibition. All the data demonstrating the updates to the PCR master mix and the validation of pooling for smegma samples has been drafted for publication, and we're planning to submit for publication in the near future. I'd like to acknowledge the many people involved in bringing this project to fruition over the years, including our molecular diagnostics team at the laboratory, and in particular, several people who were instrumental in the work, including Dr. Mankey Boonpang, who was the visionary and brainchild for this project, Dr. Tom Haregrove, who collected and provided samples for the initial validation, and Mrs. Carly Ginter Summerell, who performed all the initial validation experiments. If you have any questions, please feel free to ask during the live Q&A or reach out via email to either me or Dr. Pam Farrow, who is the section head of our molecular diagnostics team. Thank you.
Okay, thank you, Megan, for your presentation. That was very informative. We have a quick poll question for you, and then we're going to jump into our Q&A session. Okay, now that we've finished our poll, let's go ahead and jump into some Q&A. We have a couple of questions, and Megan Schroeder, we have you to answer. So Megan, what is your favorite improvement with this new reagent?
That's a good question. Actually, it's a couple of things. So first, I'd say that it's three components when making the master mix, which is really nice. It's easy for technical staff to use in the laboratory because it's just your master mix with all the components in it, your primer probe mix, and water. Makes it very easy for putting things together without making a mistake and forgetting to add something. The other thing is that I like about it is, for me personally, in our laboratory, we do a lot of homegrown stuff, so to speak. So I'm doing a lot of QC for our primers and probe mixes when I make them, and that really takes the burden off of me of having to do that because now that's on thermo, so I can get it and I know it's been QC'd and everything's taken care of. And then of course just the sensitivity of the test for detecting trick, I really like.
Oh, that's great. Just a quick reminder, anyone who's on the presentation, feel free to add your questions to the Q&A box, and we'll continue to answer those questions as well. So how many tests, how many T. fetus tests does your lab run per year?
For real-time PCR, we're looking, I was just looking at these numbers yesterday, upwards of 46,000 tests a year. When you combine that with culture, we're running about 3,700 tests for bacterial culture. So in combined total with real-time PCR and bacterial culture, we're looking at about 50,000 plus tests a year.
Wow, that's a lot of tests.
Yeah, just a few.
Yeah. We have another question. What is the biggest difference in using the FBS instead of the in-pouch?
The PBS. So a couple of things. Number one, the turnaround time, because using the PBS, you don't need to incubate. So the submitter, the veterinarian, can submit the sample in PBS, send it to the laboratory, and they can get results. If they're really in a rush, same day, but usually it's next day, just the way everything works with sample receiving and check-in here at the laboratory. But there's no incubation required for pouches. It's less hassle just for the laboratory, because we're not having to deal with those pouches. Sometimes they get really bloated and distended because of bacterial overgrowth, and they can smell pretty bad. We've had some blow up in the incubator, so we don't have to worry about any of that anymore with using the PBS submitted samples, and that's quite nice.
Wow, yeah, that sounds like it would be quite a mess.
It can be.
And how often is the T. fetus testing done? Is it an annual thing? Is it monthly? Or can you give us a little bit of
Sure. Pretty much annually. It runs through cycles or seasons. So for the molecular department for PCR testing, we start to see an increase about October, November, through usually March, April. We'll continue to run testing throughout the year, but the bulk of it's usually, I think, before breeding season, wherein you see a whole lot of testing. It tends to really be usually around Christmas time. The holidays is when it really picks up.
Oh, that sounds like good timing.
Yeah.
All right. Let's see. Do we have any other questions coming into the chat? One question I have, with the T. fetus testing, are you doing it multiple times throughout that breeding season with the same bulls, or is it just basically at the beginning of the start of the season?
To my knowledge, it's basically just at the start, I think, before they put them out. Now, if there's any question, obviously, and if the bull comes up positive, they have to go back and retest if they choose to do that instead of culling them and sending them to slaughter. But usually it's just that once that I'm aware of.
Okay. Looks like another question came in. It says, I know you mentioned 50,000 tests, but how many reactions do you especially get when you do pooling?
Oh, when you pool, let's see, do I have those numbers? So right now it's looking like about 40%. This is talking just in the pouches and the cultured samples. About 40% of what's submitted want pooling. So that will obviously, if you can pool up to 5 animals per pool, that will decrease the reactions a bit, because you're not running individually, right? So you're going from 5 individual reactions to 1 pool. Now, if that pool comes up positive, you have to go back and retest each of those samples within that pool to determine which animal was positive.
Okay.
That answer that question?
Yeah, I think so. All right. We'll wait just a second and see if there's any more questions that come in. And you mentioned that this testing usually goes through March,
April. I mean, we run it year round, but the bulk of it will be usually the beginning of the year. end of the year into the beginning of the year is what for us.
All right. Very cool. All right. Well, I'm not seeing any other questions coming in. Do you have any final remarks, Megan, before we close?
No, just thank you. Thank you for listening. Thanks for the collaboration and all the assistance in getting this project moving forward and validated and verified and in use.
Well, thank you. We really appreciate all of the hard work that you put into this.
Thank you all very much.
All right. Thanks, Megan.
Bye.